So in a previous video, we introduced toll-like receptors, TLRs, and these are receptors found on many human cells and sometimes inside many human cells, and they allow for the body to recognize molecules that are often non-self molecules. We talk about these receptors being part of the innate immune systems because they can detect and react very quickly, and the cell can respond very quickly. So the example we talked about were macrophages having toll-like receptors on their surface, in their membranes, or in vesicles. And these receptors, when they engage their ligands, they will send a signal into the cytoplasm and into the nucleus of the cell, get the cell to turn on genes and make proteins such as cytokines, which can signal to other cells locally or distally to provoke an immune response. So recall that you're going to find a lot of different types of receptors in the immune system. For example, on a macrophage, you might find phagocytic receptors, which would be used to bind ligands on pathogens and phagocytose the pathogens. Those are different than toll-like receptors. They're not Toll-like receptors aren't used to engage pathogens for the process of phagocytosis. They're used for the process of detecting a pathogen's presence, and then that cell can respond accordingly. So in this video, we're going to learn about different TLRs and how they function, and how they recognize what ligands they recognize. So, toll-like receptors are often found on many immune cells. They're also found on some non-immune cells. So again, it's just a mechanism by which pathogens can be recognized. So receptors have ligands, and in the case that we're talking about here, when we're discussing immunology, is we're talking about ligands found on the surface of pathogens or released by pathogens. So pathogen molecules. And sometimes these are referred to as PAMPs, pathogen-associated molecular patterns, because a lot of these proteins have or molecules, I should say, because they're not all proteins, have sort of this repeating pattern in them, and that allows for the toll-like receptor ligand binding domain to interact with these ligands. So let's see some examples. Before we start seeing examples of toll-like receptors, we have to recall that they act as dimers. So what's a dimer? It's two things together. And so these toll-like receptor proteins act in twos. And either the two things will be the same, so we'll talk about homodimers, or they're going to be different. They're going to be different proteins, and we would call them heterodimers. So we'll see that shortly. Let's start with toll-like receptor 4. Toll-like receptor 4 protein um, works as a homodimer. So in this one, my drawing here, I drew two TLR4 proteins, and they are dimerizing to form this receptor, this toll-like receptor. So these are both TLR4 proteins, so we call this a homodimer. The ligand for TLR4 is lipopolysaccharide, LPS, which is a molecule produced by gram-negative bacteria. So if there's a gram-negative bacterial infection, that infection can be recognized by the body using toll-like receptors. And a number of cells express TLR4 and will recognize that LPS, signal into the cell, and the cell will respond often by producing cytokines. If it's a macrophage, there are other cells that express TLR4. TLR5 also acts as a homodimer. So these are two TLR5 proteins together, and their ligand binding domain will bind the protein flagellin. Flagellin is a protein found in flagella of bacteria. So bacterial infections, specifically bacteria that have flagella on them, will be recognized by immune cells or non-immune cells, whatever cells that express TLR5. And those cells will respond accordingly, depending on what the cell is. TLR1 and TLR2, okay, these are going to interact as a heterodimer. So one protein is made by the TLR1 gene, one protein is made by the TLR2 gene, right? So these two different proteins come together and they form a heterodimer, and the ligand binding pocket of that dimer actually fits many different pathogenic molecules. So these are often found made by or on the surface of bacteria or parasites. There's quite a range of molecules, lipopeptides, uh, sugars, um, all sorts of other molecules that are often found in bacterial or parasitic infections. So, a wide variety. The last one we'll talk about on this slide here are, are pathogenic molecules that engage the TLR2, TLR6 heterodimer. So TLR2 can participate in different heterodimers. TLR2 can bind with TLR1, and that makes a certain binding pocket. TLR2 can bind with TLR6, that makes a different binding pocket. So that binds a molecule called lipotachyonic acid, found on the surface of or made by gram-positive bacteria, also made by some fungi. So again, these are recognition molecules. So the cell has recognized a pathogen, and the cell can respond accordingly, depending on what kind of cell it is. Could be a macrophage, could be a dendritic cell, could be a granulocyte like a mast cell or a neutrophil. All right. So those are some ways that TLRs are used to detect infections. You will notice on the slide here, when I'm drawing this sort of green cell, all of the TLRs I drew were on the plasma membrane, right, pointed outward into the extracellular region. So these toll-like receptors are found on the plasma membranes pointing outward, detecting pathogens in the extracellular region. But pathogens can also enter the cell. So, what about having a mechanism to recognize pathogens within the cell? So, toll-like receptors are going to do that as well. So, how can a pathogen get inside the cell? Well, pathogens can come inside via phagocytosis, receptor-mediated endocytosis. So, you can see here maybe a virus, maybe a bacteria comes in. Um, and that could be through the normal process of macrophages or dendritic cells trying to bring something in. Also, some pathogens have evolved ways to engage human host receptors and go into the vesicular system of the cell. And that's part of their um, infection processes as part of their process by which they enter the cell, especially viruses. They need to get into the cell to replicate. So viruses and bacteria sometimes actually like being inside the cell in the vesicular system, so that they can hide, so that they can reproduce. So we need, it would be nice to have mechanisms to detect them in here, and we do. So in vesicles within the cell, um, vesicles can express toll-like receptor proteins. So here I've drawn TLR7, which acts in a homodimer, and TLR8, also acting as a homodimer. So their ligand binding domain faces into the lumen of the vesicle, where the pathogen might be hiding or uncloaking, if it's a virus, for example, trying to replicate. TLR7 and TLR8 both bind single-stranded RNA. Now, what kind of pathogen would have single-stranded RNA? RNA viruses, and there are a wide variety of RNA viruses, like coronaviruses, that have single-stranded RNA as their genome or make it as part of their life cycle. So if a pathogen has come inside the vesicular system of the cell and there's single-stranded RNA in that vesicle, TLR7, TLR8 can engage them and send a signal into the nucleus of that cell, provoking some sort of inflammatory or immune response. TLR9 acts as a homodimer, and its ligand is double-stranded RNA. And again, some RNA viruses have their genome as double-stranded RNA or produce double-stranded RNA in their life cycle. So again, another mechanism by which pathogens in vesicles can be detected. One more toll-like receptor complex, TLR3, acting as a homodimer. Its ligand is actually DNA, not RNA, specifically unmethylated DNA. And it'll bind to cytosines and guanines in that DNA. Again, unmethylated DNA. Uh, that is different than human DNA, or human DNA is often methylated. Bacterial DNA, viral DNA from DNA viruses is typically unmethylated. So if if there's a virus, like a DNA virus or a bacteria, has entered the cell and released its genome, that genome can be detected using TLR3. And again, if TLR3 detects binds that ligand that can send a signal into the nucleus of the cell and get that cell to have an immune response. So these are some examples of toll-like receptors and the ligands that they recognize and where these TLRs can be found.